Hello everybody, today we are gonna talk about biology. This is Tree 3, finally the last episode. This one is about photosynthesis, which trees do, and other plants do it also. Now let's get into it. Firstly, photosynthesis is basically tree eating CO2. Tree or any plants using the sunlight to forge CO2 and H2O, which is water, into glucose, which they can use for cell respiration. So basically, like that. Now you can see 6 oxygen molecules are also produced, and now that plant can use the oxygen to do cell respiration while also using the glucose as well. But these oxygen molecules are some are left over, and now they can just to those leftover oxygen, the plant can just leave it fly away, and now the animals can use that to breathe and stuff too. And now let's get into more. Let's get more deeply into how this whole thing works. Now we have to know what carbon fixation is, and now carbon fixation is basically taking a CO2 molecule from the air and attaching the carbon to organic molecules. An enzyme called ribisco does that, and it turns RUBP into 3-phosphoglycerate. Normally, enzymes, they work at like at least 100 molecules somehow changed per second. They are very fast, but this ribisco fellow is quite slow. It's very big and very slow. And this ribisco fixes 3 carbon per second, which is very slow for the enzyme standards. So plants make loads and loads of ribisco enzymes to make efficient photosynthesis able to happen. And now we are going to talk about chlorophylls. Chlorophylls are inside well, more like on top of, let's say this is a cycloid. A cycloid is inside of a chloroplast, which is, if you look at a plant cell, a green plant cell, which does photosynthesis, if you look at it with a microscope, you can see little green blob thingies, and that's the chloroplasts. And inside the chloroplast, let's say this is a chloroplast, there are more blobs called cycloids. The cycloids fill the chloroplasts, and on top of the cycloids, you can see little frying pan thingies. It's more like a net, and that is called chlorophyll. And those chlorophylls are green. They don't use the green wavelength light to produce energy and to take energy from the sunlight and stuff. So they just they don't use the green light, so they just make it ricochet off the off themselves, and that's why the chlorophylls look green. And that's because these cycloids are covered in chlorophylls, and because the chloroplast is full of cycloids which are covered in chlorophylls, they look green, and that's why plant leaves and stuff are green. And now inside a chlorophyll, in the middle of the a more rounded part, and chlorophyll looks a bit like this. Actually, I'll put a picture up in the screen. If you look at it, it looks a little bit like a net, and the handle part is, I mean the net part, a little bit wide. That part in the middle, there is a magnesium, a magnesium atom, and when the sunlight hits that magnesium atom, the magnesium loses an electron because of well, the sunlight hit magnesium's electrons, and that made that electron have more energy, so it jumps up. And while jumping up, so when it jumps up, most of the times when an atom is hit by light, and so the electron has to jump up, it just normally loses that energy quickly, and then comes back into its original spot. But in this case, that electron is transported into a electron transport chain, which is a bit like this. This is a cycloid, and I drew it bigger, and here are the chlorophylls. And sunlight hits the chlorophylls, and now they give out electrons. Now the electrons they jumped away, so it goes like that. And the electron jumped away. So now this chlorophyll molecule is positively charged. The fact that it is positively charged means that now this molecule wants an extra electron because, well, it's positively charged. But this molecule that is called chlorophyll chlorophyll plus, because it's positively charged, this wants an electron so bad, the pull is so intense that it can pull an electron away from oxygen, which is also very good at pulling electrons from other molecules and stuff. It takes a electron from oxygen, which is in water. So basically it takes an electron from a water molecule. And so let's draw one over here. So one electron goes in there. Wait a second. Well, so if one chlorophyll loses an electron, one electron goes into that chlorophyll from water. And while doing that, one hydrogen atom gets also released from that water molecule. And so that will make one proton leaving the water. So if this is happening, so if this repeats 4 times, that will produce 4 protons and 2 oxygen molecules. Now the electron is moving towards the proton pumps, the hydrogen ion pumps. So now if this is happening a lot, there's gonna be quite a bit of protons. Oh wait, I drew one in a circle. These guys are gonna be hanging about. So as the electron passes through the pumps, these hydrogen ions or protons, they're the same thing, they are now pumped into the pumps because an electron has just gone whizzing by, and that gave the pump energy to pump the protons into the cycloids. Yeah, now these things are inside here, and now the electron ends over here. And over here where the electrons end up in, there are more chlorophylls. Sunlight also hits over here, and that would make some other electrons jump. This would jump the other way, this way, and now so there's a electron gap in those chlorophyll molecules, and this electron jumps towards here and fills that gap, and that keeps happening. It keeps rotating and stuff while producing oxygen and protons. And now it goes over here, and over here is an enzyme called reductase. The reductase reduces NADP plus into NADPH, like that. And there's also over here, more on the left side, there is also a ATP synthase. And well, the protons were pumped in, right? So now there's protons inside, and now the protons are pumped out through the ATP synthase. And I drew it backwards. It's supposed to be a little bit like this. And now the protons go through the ATP synthase, and that produces ATP, which plants can use for all sorts of things, because ATP is an energy thingy. And yeah, so through that, NADPH has been made, and also some ATP has been made. And now let's erase this. Basically, some ATP has been produced and some NADPH has been produced. So now let's talk about the Calvin cycle. This is another very important cycle thingy, like the Krebs cycle. And now, firstly, it starts off with 3 CO2. 3 CO2 molecules now come in from there and meets some Rubisco enzymes. Let's say those are the Rubisco enzymes. And also, they meet 3 RUBPs. I don't really want to draw them, so I'll put a picture in the screen. These Rubisco enzymes turn that RUBP into 6 phosphoglycerate molecules. I don't think I'm going to draw that either, so I'll just keep putting pictures in the screen. And well, that is produced, and then here's the phosphoglycerates, and now 6 ATPs and 6 NADPHs are turned into 6 ADP and 6 phosphate molecules for the ATP, and 6 NADP pluses and 6 protons are made. And through that, energy is released, and this molecule, the phosphoglycerates, are turned into GAP, or 6 GAP, or 6 glyceraldehyde phosphates. It's a hard thing to say. And now, well, these GAPs now are produced. I think I'm going to draw this one over here. There, it looks a little bit like that. And now this GAP, there are 6 of them. And one of these molecules, one of these GAPs, are thrown out. Well, not thrown out, moved into a different place. So now there are only 5 of them. Those 5 GAPs are now turned into 3 RUBPs, while 3 ATP molecules are being used to make energy and phosphate molecules. A little bit of phosphate molecules stick onto the GAPs, and a little bit of changing happens. And as the whole thing happens, 5 RUBPs are made. So, and now, well, on the first bit of this cycle, 3 CO2 molecules and 5 RUBP molecules combine through the Rubisco enzymes and turned into phosphoglycerates. So basically, this cycle now can repeat again. Now the Calvin cycle can let 3 more CO2 molecules in, and this whole thing happens again. And each cycle, one GAP is made from each cycle. And now, so we get GAPs now, and now we're gonna do some more stuff with the GAP. Now, 2 GAP molecules combined, they have 6 carbon atoms in it combined, and so 2 GAP molecules can forge into one, and that way produce glucose. So that's how plants store glucose in their whatever they are storing glucose in. Maybe it's their berries or fruits, roots, or whatever. They produce glucose by forging 2 GAP molecules into one. And also, they could use that glucose for respirating. But before that, if you watch my other video, Tree 2, where I explained the Krebs cycle and a little bit more after that about cell respiration. Actually, I might have did it in the first video, Tree 1. But while cell respiration happens, it turns glucose into 2 GAPs, and then turns the GAP into a molecule called pyruvate, and uses that pyruvate to run the Krebs cycle, and uses that pyruvate to make acetyl groups, and produce a CO2 molecule which flies away, and that acetyl group runs the Krebs cycle, and that way ATP is produced. So, basically you need GAP to run the Krebs cycle, so why need to turn the GAP into glucose and then break it down again and turn it into GAP again and run the Krebs cycle? You don't need to do that to produce ATP if you just have the GAP molecule right there. So now what the plant does is it doesn't turn it into glucose. It just uses that GAP and runs the Krebs cycle. Well, after turning it into the acetyl group, but it doesn't forge it into one. It doesn't need to, and well, that's how they get the ATP they need for being alive. Yeah, as I said, some leftovers are forged into one and make glucose, so the animals can eat it and survive. And now, this is how photosynthesis works. I think this is all I really needed to say. So, bye.